So today we're going to be talking about some of the main methods for studying the brain and behaviors. So first, a quick overview of the types of studies. There are 3 of them, and they're descriptive, experimental, and correlational. And an interesting type of descriptive study are known as case studies. And case studies usually have a limited sample of participants, and because of this have low generalizability. And they also take a deep dive into something unusual or unique. So to the right, you can see Phineas Gage, who had some damage to his prefrontal cortex, which led to both changes in behavior and cognition. Another example is a man named Tan, and he was called this as he could only say the word tan after damaging Broca's area. Another type of study are lesion studies. In this video down on the bottom, it's an example of lesioning the hippocampus of mice, which led to problems with spatial memory. And when they're experimentally done, they are done with animals, as it would be quite unethical otherwise. And lesion studies are very good at helping scientists determine the functioning of different parts of the brain.
So when it comes to brain imaging and mapping, scientists have a variety of different technologies that they can use, and all of these technologies image specific phenomena. So the first one is magnetic resonance imaging, where scientists use a strong magnet to align the protons in water molecules. And after the magnetic field is released, the proton spin returns to normal. So the scientists use that information about the rate at which the protons go back to normal to differentiate between soft tissues like white and gray matter. Unfortunately, MRI scans can take hours, so the temporal resolution is pretty bad, even though the spatial resolution is really good. fMRI operates on the same principles as regular MRI, but instead uses those proton spin calculations to measure the oxygenation levels of blood. So the principle behind this is that areas of the brain that are more active are also going to have more highly oxygenated blood. And again, it has really good spatial resolution and not that great temporal resolution. Positron emission topography uses a radio-labeled ligand to detect which parts of the body have higher levels of metabolic activity. So similar to fMRI, parts of the body with higher metabolic activity are going to be more active. As you can see in this picture, more active, less active. Again, higher spatial resolution, low temporal resolution.
So as you can see on the image on the left, during an EEG, sticky electrodes are placed onto a participant's head. And since neurons are polar cells during an EEG, changes in charge separation and the movement of charged particles in response to behavior are measured. And this form of measurement also happens in real time, which means it is great temporal resolution with a trade-off for a lower spatial resolution. And EEGs are great for studying order of operations and signal magnitude. And then computed tomography, or also known as CT scans, they're great for visualizing the brain structure. And it's used to diagnose things such as brain bleeds and tumors. And the way this works is that multiple 2D X-rays are stacked on top of each other to create a 3D structural model, as you can see in the right. And this technology is non-invasive, but it does involve undergoing radiation.
So chemical signaling methods are another more broad category of experimental and correlational studies. And generally they allow for more accurate and clear results and great analysis of temporal and spatial correlation. However, they are more invasive than the other forms of studies. So in vivo microdialysis is one form of chemical signaling that measures neurotransmitter release during brain activity, and it provides great spatial activity analysis. It also allows for the collection of samples in living, free-moving animal models rather than post-mortem tissue. The samples that are received are from interstitial spaces of the brain where neurotransmitters are present, and one of the only methods that provides quantitative study activity is in vivo microdialysis. So the picture on the bottom left shows the overall methodology using technology as well. The picture on the bottom right shows perfusate pumped into the brain and dialysate pumped out of the brain, which is the sample that's analyzed. And the picture to the top right is an overview of how in vivo works. So spinal taps is one other form of chemical signaling. It's also known as lumbar puncturing, And basically, it measures circulating levels of neurotransmitters throughout the body, and it's generally used in the clinical setting rather than the research setting. So a balance of neurotransmitters is measured for overall well-being of the patient, and the physician measures CSF, which is cerebral spinal fluid, and they can analyze pressure to detect if hydrocephalus is present in the brain. Infections in the brain are also diagnosed with spinal taps, and in general, they're less invasive and less risky than procedures on the brain. So the image on the bottom left shows where exactly the spinal fluid is extracted from, and the image on the bottom right shows the steps that a physician would follow when conducting a spinal tap. So thank you so much for watching our video, and I hope it was informative.